recording. So this is, I think this is topic 1.6 maybe. This is mitosis. Okay, so in mitosis we have 4 phases. The analogy I use for mitosis is a play.
What?
There's 4 acts. My analogy for this whole entire thing is you have 4 actors and you have 4 acts. So act 1 is prophase. Act 2 is metaphase. Act 3 is going to be anaphase. And act 4 is going to be telophase. My actors, I have 4 actors. Or I should say thespians. Keep it We listening? All right. So my 4 actors is going to be chromosomes, the nuclear membrane, remember I'm recording this. All right, nuclear membrane. I don't know how sensitive the mic is, so I might pick you up. So chromosomes, nuclear membrane, centrioles, and spindle network. Spindle fibers or spindle network. It's the same thing. Okay, so these are my 4 actors. Chromosomes, nuclear membrane, centrioles, spindle fibers. Act 1 and Act 4, you're going to use all 4 of them. So in Act 1, when I have prophase, so in prophase, what is each person doing? So the chromosome is unraveled. So the chromatin is a chromosome, but it's not wound up. And so during prophase, the chromatin is going to become chromosomes. So you can use a few different words to describe this. You can simply say, they condense. You can say chromatin condenses into chromosomes. You could say it just simply winds up. Doesn't matter, right? The chromatin winds up into chromosomes during prophase 1, during prophase. Okay? The centrioles, they are going to move to opposite ends. There's 2 of them. They move to opposite ends. And the spindle fibers, they begin to form. Okay? And who's my 4th actor? Oh, the nuclear membrane. The nuclear membrane dissolves. Okay? So if I'm going to draw this, you start with your cell, draw a nice big circle, and then on the one end, make like a T. So your centrioles are normally drawn like a T. Right? And then draw your nuclear membrane and draw it dissolving. And in the membrane, you can draw them as X's if you like. You can draw them as X's, or you can just draw squiggly lines. So your chromatin is becoming chromosome. That's how you draw a prophase. Centrioles. The centrioles are the red squares are the centrioles, and they're going to move to opposite ends. So this picture actually might be early prophase. So let's say this is early prophase. So this is early prophase. If I look at like late prophase, the nuclear membrane might be all gone. The chromatin is definitely shaped like chromosomes. So there's no more chromatin, right? And here's my cell, and then my centrioles are on opposite ends, and the spindles begin to form. So this is like early versus late prophase, right? Early prophase. So, like I said, this is Act 1 of the play. Act 1, what's the nuclear membrane doing? All of these will reappear in Act 4. Just like a lab report. Whatever you say in your introduction, you say in your conclusion. When you write an essay, right? Whenever you write an essay, right, you start at the beginning and you introduce it, and then you have a conclusion. So think this the exact same way. So this is prophase. Now so go to Act 2. So Act 2. So metaphase. The nuclear envelope is not there. So there's no nuclear envelope or nuclear membrane. So there's my cell. The chromosomes, where are the chromosomes?
Line in the middle.
Right, lined up down the middle. So my chromosomes are lined up down the middle. My centrioles are on opposite ends. And the spindle network is fully formed. And the spindles are attached to the centromere of the, so on a chromosome, okay, so real quickly, a chromosome, here's my chromosome. Each half of the chromosome, these are called sister chromatids. And the center of this is called the centromere. And the ends of the chromosome, these are called telomeres.
Do you have to know that?
Yes.
Sort of.
That's a yes.
Telomeres, what you should know about them is that telomeres, cancer cells, as your cells get older, your chromosomes lose DNA and your chromosomes become shorter. So as your chromosomes, so it's called telomere shortening. So as your telomeres get shorter, you are higher chance of getting cancer, and this happens with age. So my chromosomes are shorter than your chromosomes. So that's all we need to know about that. So sister chromatids, centromere, telomeres. So that's metaphase.
Can you like repeat it?
Yeah, I'll go through it again. Yes.
The telomere grows again or never grows back?
Well, no, like once, because what's happening, what we think is happening is that when the cell divides every time, that some of that DNA gets cut off, and if it gets cut out, it doesn't get back in there, right? So over time, your chromosomes get shorter. If there's a case of, there could be some case of somebody somewhere where the chromosomes get longer, but it's probably, remember, every time your cells divide, it has to copy all the DNA. So there's always going to be some DNA that gets cut out. Maybe some gets added somehow because maybe another chromosome broke off and got added to another chromosome. I'm sure that happens too. So just imagine you have to copy all of your chromosomes every time. There's always going to be some mistakes, and some chromosomes get cut and hacked off. And in general rule is they get shorter over time. Okay? Telomere. MERE. Centromere. So easy to remember, they're both meres. Telomere, centromere. So telomeres, centromere, okay? So one more time. Who are my actors? Nuclear membrane, chromosomes, centrioles, spindle, right? So in metaphase, the centrioles are opposite ends. The spindle fibers are fully formed. And the spindle fibers are moving the chromosomes around the cell. So the chromosomes are lined up along the equator. What act am I on?
Act 2, 3.
Act 3. So anaphase.
Come here.
And Bonnie, this is why we have to know what sister chromatids are. So the key word in anaphase is the sister chromatids. the sister chromatids are pulled apart at the centromere. That's the key action in anaphase. This is the climax of the story. This is the climax of the play. Because if you have non-disjunction, if chromosomes don't move equally, you have non-disjunction. So how do we draw anaphase? You can draw your cell a little more elliptical. So instead of making a circle for your cell, draw your cell a little more elliptical. You still draw your centrioles. Still draw the centrioles. Your spindle fibers are not as long. Your spindle fibers are shorter. And the chromosomes don't look like X's, they look like V's because they're pulled apart. They're pulled apart at the centromere. So this is your anaphase. So your sister chromatids are pulled apart at the centromere. Okay? Your cell looks a little more ellipsoidal. Your you still see spindle fibers, you still see your centriole, and your chromosomes look like V's, not X's. Okay?
Yep.
What do you need to see?
Yes.
The sister chromatids are pulled apart at the centromere. All right?
At the same, from the same.
From the central.
At the centromere. Or from the, doesn't matter. It's just English.
From
Yeah, English A.
Right? I mean, I believe if you on any question, if as long as you said during anaphase, the sister chromatids are pulled apart. You don't want to say the chromosomes are pulled apart. And when we talk about meiosis, in meiosis, the chromosomes pair up. In mitosis, they don't. Because mitosis, we're making a copy of a cell, so we don't care. We take all the chromosomes, we copy them, and split them in half. Right? In meiosis, they have to pair up, and the sister chromatids don't split until division number 2. In mitosis, they're pulled apart in the first division. So we'll get to that next. So Act 4. Now your Act 4 is telophase. Now your cell looks like a peanut. So you can kind of draw. It's called the cleavage furrow. So you can draw kind of like basically draw 2 cells now. So you want to draw 2 cells, and you show the cleavage furrow. Okay, so you can simply draw it like a peanut. Cleavage furrow. Okay, now who's here? Is the spindle fibers have exited stage left? There's no spindle fibers. The centrioles, they're no longer at the equator. Or sorry, there's no longer 2, right? There's one centriole here, and there's one centriole on this side, on each side of the cell. What's the nuclear membrane doing? Reappearing, reforming, reappearing, building back up again, whatever you want to say, but it's coming again. So now you have your nuclear membrane, right? And is it chromatin or chromosomes? Chromatin. So you can say the chromosomes are unwinding. So I like to draw chromatin like squiggly lines. So you can just draw chromatin like that. You could simply just say chromosomes are unwinding.
You drew it like that last time.
Yeah, you could also draw it. You could draw it like this, or you can draw it with actual chromosomes. So if you want to draw it with, I'll erase this. So if you want to draw it with squiggly lines, or you want to draw it. But remember, the chromosomes in this are not X's. The chromosomes in this at this point are going to look like staggered V's because they're only X's when they're before they divide. When a chromosome looks like an X, it's because they've been copied. So one arm of one side of the X is identical to the other side of the X. All right. This is mitosis. This is nuclear cell division. the cells are exactly the same at the end. So we have Act 1, Act 2, Act 3, Act 4. We have 4 actors, centrioles, chromosomes, nuclear envelope, spindle fibers. All right? Now, we got 5 minutes left. Do you want to try to fit in meiosis? Because just the differences. If you know mitosis, how is it different? So, we have mitosis on one end and meiosis on the other. The first step. Okay, so in mitosis, you form 2 identical cells. In meiosis, you are going to have 4 unique cells. So, in mitosis, how many divisions are there? 1 division. In meiosis, you're going to have 2 divisions. So we're not going to have an S phase. There's no interphase. There's no interphase between meiosis 1 and meiosis 2. Okay? So the key differences between mitosis and meiosis is that you have prophase 1. In prophase 1, you have homologous chromosomes pair up and form. There's 2 words you can use, tetrads, or you can call them bivalents. Either one is acceptable, and you'll see either one used. You're more likely to see bivalent used on a test. If you're revising and watching videos online, I bet you're more likely to hear tetrad than bivalent. Okay? So if you're watching a video to review, you might hear a tetrad. Because you have 4 arms, right? If you have two X chromosomes line up, now you have 4 arms. Okay? And during prophase 1, this is where we have what's called crossing over. So during when the chromosomes pair up and form tetrads, we have an event called crossing over. And this is, in other words, is genetic recombination. So the genes shuffle, and they shuffle up. And then it's in Anaphase 2. So in Anaphase 1, the homologous chromosomes. So okay, that's one key difference. Next key difference is Anaphase 1, the homologous chromosomes split. And then Anaphase 2, the sister chromatids split. I'm getting sloppy here. We're almost out of time. So I'll upload this for you guys. Homologous chromosomes. So those are your key differences between mitosis and meiosis.